All right, so we're going to have an activity in order to go over how you phenotype bacteria, and I know this will be a little bit challenging for everyone. And now to make it a little bit more complicated and more fun, we are also going to think about gene transfer between bacteria. So this will require both thinking about how DNA can be exchanged between bacteria, how it can end up into another chromosome, and then also how you phenotype. So gene transfer, vertical gene transfer or horizontal gene transfer. So, when we have gene transfer in humans, typically the only way we can share our genetics, or if we have a new allele, for example, that arises within our genome, the only way we can actually pass this on is through vertical gene transfer. So this is gene transfer of genetic information between members of the same species. And so I can pass it on, for example, to my offspring. Bacteria are able to do horizontal gene transfer. And so this is pretty powerful. So this allows for genetic information or DNA sequences, for example, particular alleles, new genes. They can actually transfer this information between related but distinct species. And so they don't actually need to pass this on either in a vertical direction, either through their offspring, and that also means that they don't necessarily have to receive this through their parents. And so this is powerful, and unsurprisingly, it plays a significant role in the evolution of bacteria. So it's a big reason why antibiotic resistance spreads so quickly, is that if a bacteria becomes resistant to a given antibiotic, you're not limited to that species. That gene can actually transfer between a number of different species of bacteria. And so that allows us to spread much more rapidly than normal. And so how does genetic information exchange between different bacteria? And there's three primary modes that we're going to talk about. So conjugation, and there's actually two different ways we're going to talk about conjugation, but they both utilize the same process. And that is where DNA is actually exchanged across a bridge called pili. And so one donor cell is able to send DNA into a recipient cell through this bridge. And this can either be a plasmid, and so the reason we distinguish this into two different cases, it can either be a plasmid that gets sent in, or it can be the chromosome that actually gets sent in. And what actually gets transferred has a very different outcome on the bacteria. You can also use transformation. And so transformation is when DNA in the environment makes its way into a bacteria and it is able to uptake that into the cell. And so typically the donor cell, so in all these cases, the middle cell represents the donor cell. In this case, usually the donor cell has died, it is lysed, and it's released part of its chromosomes into the environment. And then some other random cell nearby can actually take that DNA up. And then the final way is actually utilizing bacteriophages. And so if a phage is able to transfer DNA from the host cell into a recipient cell, that is the process is called transduction. So part two, we're gonna first talk about conjugation. And conjugation was discovered through study of a factor called the F factor. And so the F factor stands for fertility. F stands for fertility. And so this allows for the unidirectional transfer of genetic material. So a little parlance, F plus cells serve as DNA donors. F minus cells are the recipients. So in this case, the way we've drawn this here, this would be the F plus cell right here. And then this would be an F minus cell. And then in this case, this would be an F minus cell. And so they confer the ability to donate DNA during conjugation, and that is because they're able to actually construct these pili. So F plus cells are able to build these pili bridges, and these pili bridges act as one-way channels where DNA can go through these pili. And so the reason they're F plus cells is they know how to build these pili and send DNA into the other cells. So these cells, these F- cells, they don't know how to build the pili, and they don't know how to send DNA into the other cells. So they can only receive whatever is sent in. So E. coli may or may not contain the F factor, and if the F factor is present, one way you know that is if you look underneath a microscope, you can see these cells have the sexed pilus. So pilus is the singular, pili is the plural version of that. And so this serves as the donor of genetic information. This is how the DNA can actually make it from one cell to another. However, F factors also have this remarkable property that if an F plus cell is able to undergo conjugation with an F minus cell, the F minus recipient is converted to the F plus state. So it's kind of like a virus almost, like the Matrix, where like once a bacteria cell is infected, it becomes an F plus as well. And so why is that possible? So it's possible because the F factor is actually a plasmid. So the reason these cells are F plus cells is because they contain a plasmid that contains genetic instructions for how to do all this stuff. So in part, this F factor contains all of the genes necessary for building these pili. So I don't know if I have a picture of this. So a bacteria cell will actually have kind of multiple pili sticking out. And so these pili are all constructed out of like unique proteins. And there's actually a large number of proteins that kind of go into building this pilus. So this plasmid contains the genetic instructions for all of that. This F factor also contains information for transferring DNA into the cell. So this F factor creates these pili. These two cells come near with each other, and then this bridge is able to form through the pili. And so now you have to be able to get DNA to actually transfer through this. And so this F factor actually contains a small piece of DNA called the origin of transfer. And this origin of transfer is recognized by proteins kind of at the base of the pili, and it's able to actually funnel this into the other cell. And so it's not actually both strands of the plasmid. You actually have a nick that occurs, and then one of the two strands of the plasmid is moved across the conjugation tube. As this is happening, DNA is synthesized. So this single strand of DNA, a new copy, a complementary strand, is formed based upon that in both of the cells. So as a result, this F factor gets transferred across, and then at the same time, DNA is synthesized, and then you ligate this together, and so you end up with two full copies of this F factor plasmid. And so this is the reason why the F factor is able to transform. It has this transforming principle, is that it's able to pass on all of the instructions that are necessary for making this cell an F plus cell. So now this cell, which was formerly an F minus cell, now this is an F plus cell, and if it finds an F minus cell, it's able to kind of undergo the same process. So, why is this important? So this is important because, these plasmids, this F factor plasmid contains all sorts of genes for making the pili, etc., but it can also have extra genes as well. So if this plasmid happened to have a streptomycin resistance gene on it, so if this gene encodes the ability to be resistant to streptomycin, then now we've not only taken this cell and made it an F plus cell, but this F plus cell is also able to be resistant to the streptomycin antibiotic. And so in that way, the antibiotic resistance is able to spread. Part of the remarkableness of this is that these bacteria don't even have to be the same species. So this could be species 1, this could be species 2, and this DNA transfer is still going to be able to happen. And so this is what gets back to the whole idea of horizontal gene transfer, is that this process does not require them to be the same species. So a human, for example, can't transfer DNA to a chimpanzee, because there's genetic barriers that prevent reproduction with a chimpanzee. But that's not true with bacteria. So bacteria are able to actually transfer DNA between different species. So this is conjugation, where we moved one plasmid from one cell to another. And now we're going to talk about what happens when we move genomic DNA from one cell to another. And so this process was discovered because there are these cells called Hfr cells. So these are a special category of F+ cells. And through a large amount of subsequent research, they figured out what happened was that in some cases, this plasmid is actually able to integrate into the host genome. And so in this case, we show the plasmid integrating between gene A and E. So there, again, bacteria have a circular chromosome, and this circular chromosome is about 4 million base pairs long. And so now this plasmid is integrated in the chromosome. It's still able to produce the proteins necessary for forming these pili. So this is still a cell that is able to undergo genetic exchange with an F- cell. However, again, because the F factor actually specifies, it has a part of its DNA that specifies what gets transferred into the new cell. The first thing that actually gets transferred is a small part of the plasmid, which is shown in kind of like blue right there. And then after that, what gets transferred is the host genome. And so now we're actually able to transfer the host chromosome into the recipient cell. And so the idea is that these cells these bacteria species are similar to one another, but there's some sort of genetic variation. And so the way we've designated this is by either having capital A, B, C, D, or E, or little a, B, C, D, and E. So you notice the host cell has capital alleles, and the recipient cell has lowercase alleles. And so the idea is that each of these cells has different alleles of these genes. And these can be recognized in some sort of phenotypic way. So you have transfer begin. And so initially, and again, it's a single-stranded, only a single strand gets transferred. And then immediately you start replicating DNA in both cells. So this gets filled in, and then this single strand of linear DNA gets sent in. So you might think that this entire chromosome then is going to be able to make it into this bacteria cell, creating a diploid bacteria. But this is not what happens. So typically, conjugation is interrupted before the chromosome transfer is complete. So this is 4 million base pairs long. A plasmid is about 10 kb long. So it's 3 orders of magnitude smaller. And so, the cell is able to transfer the entire plasmid, but it's unable to transfer the entire chromosome. And so at some point you have breakage occur. And so you'll notice that there's a host chromosome in the recipient cell, and then there's also a linear piece of DNA from the host cell. So what happens to that linear piece of DNA? So this linear piece of DNA is unable to be replicated. So a bacteria cell can only replicate circular DNA. It's not able to replicate linear piece of DNA. So normally you would think it would just eventually get lost from the cell. But the cell is actually able to recombine parts of the DNA from that linear strand with the host chromosome. So it's kind of similar to, the process is kind of similar to what happens during meiosis, where you have a double recombinant occurring where you have it go from one DNA strand to the other, and then it can kind of jump back. So if that happens, you can actually permanently transform the recipient chromosome with the alleles from the host. And this is shown here. So in one case, you have a recipient chromosome along with this blue linear fragment. And I'm showing you the case of a double recombinant, where one recombinant event occurs here and one recombinant event occurs here. And you can notice that there's 3 genes that we've shown, and there's different alleles in each. So Lu minus, Arg minus, Met minus, so these are amino acids. So this particular recipient cell is unable to live without supplying leucine, arginine, and methionine. But if this linear piece of DNA is transferred and the recombinants occur here, then you're able to actually exchange this part of the DNA. So you'll end up with a Lu plus Arg minus Met minus. So these genes will remain the same, but this gene will be exchanged. Similarly, if the double recombinant occurs here, what you'll have is the host goes over to here, and then it moves back, and then you've changed the Lu and the Arg gene. Or it can be on the left side and the right side, and then you've actually managed to exchange all 3 of the genes. So all 3 of these are exchanged, and the resulting genotype is Lu plus Arg plus Met plus. You can also have quadruple recombinants. So this is a case where you have 1, 2, 3, 4. And in this case, you would exchange the first gene, then you would move back to the original, and then you would exchange the third. So in bacteria, the quadruple recombinant is very similar to the double recombinant, from the previous chapter. So in this case, we have exchanged the first and the third gene, but not the second one. And one thing to note is that you always need to have an even number of recombinants. Otherwise, what you would create is a single linear piece of DNA. So I'll let you work that out on your own. If there's 1 recombinant or 3 recombinants, but what you would find is that instead of having a linear piece and a circular, you would only have a single linear piece of DNA, and then the cell would actually die. So there's always an even number of recombinants in these situations. So differences between HFR and F+. HFR is a special class of F+ cells that occurs when the plasmid integrates into the host genome. The HFR strain donates genetic information to the F- cell, but there's some really key differences between them. So the recipient does not become an F+ cell. And that is because the plasmid, you'll notice that the majority of the plasmid is not actually transferred into the recipient cell. So all those instructions for how to build the pili, all those instructions for how to transfer DNA, all that genetic material isn't actually transferred in the recipient cell. So the recipient cell of an HFR exchange is not transformed. F+ and F-, so the recipient becomes F+, so this is the F+, and then the HFR crossed to the F-, the recipient remains an F- cell. So this technique, using these HFR strains, has actually been a powerful way to actually do initial mapping of the bacteria chromosome in order to figure out the order of genes. And so what are these experiments? So the idea is that you can control how long these conjugation experiments go on. And so if you put these cells in a blender and then just blend them up, the centrifugal, the forces, the shearing forces that are caused by the blender spinning the liquid around will actually break the pili so that you end the transfer of DNA. And so it's a way to control, how long this DNA, how large of a piece is actually transferred into the recipient cell. So again, you have to know where the F factor has integrated, because that specifies where the initial piece of DNA gets transferred. So you'll see this little red piece right there. That indicates, it should be on this side right here. That indicates the initial part of the plasmid that's transferred, and then the DNA moves kind of in this direction. So you'll see this orange gene gets transferred first, then the purple, then the orange, and then the green. So in time order, this is the first gene that gets transferred, this is the second, this is the third, and then this is the fourth. And so this is a graph from a large number of experiments. where you've interrupted the conjugation at a specific time. So you've mixed the two strains together, you've mixed this strain together with this strain together, and then you've waited 10 minutes before you put them in the blender. Or you've waited 15 minutes, 20 minutes, etc., 25, 30. So there's all these, this is just an example of, dozens of different experiments that were done. So the original strain was sensitive to the antibiotic, not the antibiotic, to the drug sodium azide. So sodium azide is an ATPase inhibitor. And so this strain right here is sensitive. The S means that it's sensitive to sodium azide. And so the only way this cell will be resistant is if this piece of DNA actually exchanges into the genome. So you notice that occurs first. So this phenotype right here is the first one to change. Sodium azide resistance is the first one to occur, and you'll also notice it goes up to the highest amount. The next one is TON, so it's resistant to TON, which is a bacteriophage. So initially the cell is sensitive to this phage, and then as time goes on, you start to see it exchange into a resistance. But again, you'll notice that it takes longer and it doesn't go up to the same level. The lactose is the next, and then finally the gal phenotype is the last one to exchange. So again, these are two sugars. So this cell is originally not able to grow on galactose or lactose, but eventually, due to the exchange, it receives the allele that allows it to grow on lactose and then galactose. You might wonder why they all don't go up to 100%, and that's just because there's kind of like a natural but stochastic ending where even if you don't use the blender, conjugation will end. So in some of the cells, even if you wait 60 minutes, for some of the cells, it happened just to have the transfer end at this point. And so if the transfer ends at this point right here, then you're not able to exchange the sodium azide gene. When you get to galactose, like there's a bunch of random endings kind of in between, even though you've waited 60 minutes. And so, you only go up to about 25%. So this was able to show that the chromosome of Hfr strains were transferred linearly, and then the gene order was able to be predicted, and then it also was able to demonstrate a circular chromosome. So now the idea is that you take a large number of Hfr strains that each have a different integration point. So this just shows where exactly the F factor has integrated into the chromosome. And so by using that, you can map out a large number of different genes, and then they were able to show that it was circular in nature as well. So an F prime strain, so one final modification of that strain. So this occurs when the F factor can lose its integrated status. So it's able to revert back to the F plus state, which means that this integrated F factor actually excises out of the chromosome. However, this excision is not perfect, and so because this excision is not perfect, it might grab some of the host chromosome nearby. So that's really bad for this F prime cell because it no longer has potentially essential genes on its chromosome. Now it's reliant on them to be on the plasmid. But now when you transfer, when it undergoes genetic exchange, this new plasmid that the donor cell receives is actually containing DNA that's on the host chromosome. So this is a way to create what's called a merozygote, and also sometimes called a merodiploid. And so a merodiploid is a diploid for a small number of genes. So this particular recipient cell has two copies of the A gene. It has little a and it has big A. and also has 2 copies of the E gene, little e and a big E. So it's actually acting kind of like a diploid cell for these 2 genes. And so this is also important because it allows you to take genetic material from the chromosome and transfer it much easier among different cells. So for example, if this A gene encoded streptomycin resistant, now you would have a plasmid that would encode streptomycin resistance that could spread, very easily. So an F factor is an example of a plasmid. So it's a double-stranded closed circle of DNA. It exists in multiple copies in the cytoplasm. It can contain 1 or more genes, and it's hugely important for the spread of antibiotic resistance. All right, so transformation, we'll just end part 2 with just a little bit on transformation. So transformation, we talked before, right at the beginning, was the process of free DNA being taken up by the cell. And so following along with the conjugation will hopefully make understanding transformation very easy to follow. So transformation occurs because there's free DNA within the environment. So if a cell lyses, if a phage lyses a cell, or if it just dies from kind of other reasons, the DNA within the cell can be released into the environment. And so some bacteria, and this is not all bacteria, this is an active process, but if bacteria are able to take up DNA, they're called competent bacteria. The DNA is recognized by receptors, and then this double-stranded DNA that's in their environment is able to make its way within the cell. So the strands segregate, and again, you have a single strand actually degraded, and then the other strand can pair homologously. So again, linear DNA, it can't replicate within the cell, but it does have the power of recombination. So this piece of DNA could potentially recombine. So if there's enough homology between this blue piece of DNA and the orange piece of DNA, you're able to actually transform, the genes that are in common. So just think of this piece of DNA as serving the same purpose as the strand that's transferred in the Hfr cell. So outcomes of transformation, so the DNA enters into the recipient cell, and then recombination between the foreign DNA and the homologous region in the chromosome allows for a kind of a permanent transformation. So both of these steps is required for any sort of permanent effect on the recipient cell. So the first step alone results in foreign DNA in the cytoplasm, but it's only the action of recombination that allows this DNA to enter into the chromosome. And so finally, co-transformation. So co-transformation is the idea of that simultaneous transfer of genes. So if genes are close enough to be linked, they are carried together on a single segment of 10,000 to 20,000 base pair pieces of DNA. So how frequent 2 genes are transformed? So it's kind of like a process of mapping. So before we talked about map distance in chapter 5, and the idea behind that is that genes that were closer together, it was much harder to form recombinant gametes that way. And then in bacteria, the idea is that genes that are closer together, they're much more likely to co-transform. And so that means you would change both of the genes at the same time. All right, that is the end of part